Okay, my paper's on arsenic and the glucocorticoid receptor, which I'm going to refer to as GR. So the purpose of this study, I tried to summarize it the best I could, is to examine a toxin, they use arsenite, the ability of it to alter nuclear transcription factor activity. And do non-toxic levels of arsenite alter the function and activity of GR? So, just a little background about arsenic. It exists in groundwater, and New Hampshire isn't included, and this is relevant to you. And I'm from a town where everyone has private wells for drinking water, shower water, and stuff like that. So I thought this was pretty interesting. And it has been associated with increased cancer risk, including lung and skin cancer. Arsenite in non-toxic doses has been shown to alter inducible gene expression of PEP-CK gene. DEX is used in the study as a synthetic glucocorticoid. The PEP-CK gene is primarily transcriptionally regulated, and it's been studied really well, so it's like a good model gene. Its cytosolic enzyme product is a rate-limiting step in gluconeogenesis, and hormone-regulated expression is regulated by GR-mediated pathway. So the methods there's a lot of stuff, like it was talking about plasmids, it was really in-depth, so I just kind of summarized it because there's also 9+ figures, so I figured the time would be better spent on that. So they use rat liver cancer cell line, H2 or IIE cell line, and they did a transient transfection of plasmid DNA into the liver cell line. And then they used to do the Western blot, they isolated proteins and precipitated them out for a pure GR collection of proteins to use for the Western blot analysis. Then they used the FIGR antibody to determine levels of GR protein in the cytoplasm and in the nucleus of these rat liver cancer cells. Then they also used confocal microscopy, which I thought actually was like the best indicator of their results. It really showed like the whole study really well. And they used green fluorescence protein, GFP, and propydium iodide fluorescence, which is the iodide one is like a red stain, and they did this to visualize localization of GR in the cytoplasm and the nucleus. So they kind of used the confocal microscopy and the Western blot kind of side by side, and they did end up getting pretty much the same results. So the results were the arsenite treatments can reduce GR transcriptional function significantly. Arsenite may alter the PEP-CK gene expression by suppressing GR-dependent regulation of the promoter. And arsenite doesn't actually change the levels of the GR receptor in the cells, it just alters the way that it functions once it gets into the nucleus. And I'll show you about that in a second. Arsenite inhibits the expression of the PEP-CK gene and tyrosine aminotransferase GREs, the glucocorticoid response elements. So this is the confocal microscopy. So the A and D, these two are kind of the controls. So this is showing you that with green fluorescent protein staining, that all of the GR, because they did like a construct, they used green fluorescence protein and tagged it onto the GR. And so it's all localized into the cytosol of the cell. So this dark part here would be the nucleus. And then here they use the red stain as well. So the GR is still in the nucleus, it's still in the cytosol and not in the nucleus. So the red stain stains for DNA, and they digested all the RNA because it would also stain for that. So that's pretty much just to stain in the nucleus where all the DNA would be located. And then, so these were pretty much your control cells. And then in B, that was DEX alone. Yeah, so this was DEX alone. So that was the synthetic corticoid hormone. So that all localized, it brought all of tag GR into the nucleus. So as you can see, this is the cytosol, this is the nucleus. So all of it went into the nucleus. So it's showing that with DEX, the GR is able to translocate from the cytosol into the nucleus. And then in this one, you can see that the red cells were just the DNA, and then these, it's kind of hard to see on this because it lost some of its resolution, but these were like lighter. So these were overlapped, so you can see that it's still stained red, but also all of the green fluorescent tag GRs translocated into the nucleus. And then in C, this was arsenic and DEX combined. So they were trying to figure out if you have DEX, which is the synthetic hormone, and you have arsenic. Does arsenic inhibit the ability for DEX to bind to the GR to translocate into the nucleus? And this shows that it doesn't. So here is arsenic and DEX, and GR still translocates into the nucleus, even with arsenic exposure. And the same thing here. It still translocates into the nucleus. That one kind of lost results too. So arsenite does not alter DEX-induced translocation of GR. So the GR can still translocate into the nucleus. And so the control cells, I don't know if you guys can see that says control. This is arsenic and DEX, and this is DEX alone. So this is cytosolic, and this is nuclear. So they were looking at, and this is GR, and then this is their control. So in the cytosolic protein, the control, it's still shown in, the GR still in the cytosol. And then if you look at nuclear, with arsenic and DEX exposure, or DEX alone, the GR is now found in the nucleus. So DEX treatment showed GR abundance in the nucleus, and arsenite had no significant effect on translocation of GR into the nucleus. So it's pretty much just backing up what they saw in the control. Arsenite does not alter total cellular GR level. So like I said before, it's not a matter of reducing this protein's presence, it's just a matter of its function. Arsenite did not affect the ability of GR to translocate. So it's just, again, here's nuclear translocation, here's the control, here's arsenic alone, here's DEX, so more translocation with DEX exposure, and then arsenic and DEX, and it's the same. So PEP-CK expression was reduced with arsenite doses. So arsenite inhibits GR-dependent transcription via nuclear event and function once it's in the nucleus. GR is involved with glucose homeostasis, immunity cell growth, and differentiation. So it's important, like, all around for body physiology. GR exists in a preactive state in the cytosol, and then when the steroid binds, there's a conformational change and it unmasks the DNA binding domain on the GR protein. And then GR translocates to interact with DNA into the nucleus. And GR also plays a role in how arsenic acts as a carcinogen causing cancer. So GR can suppress tumor promotion by suppressing cell growth. So reducing its function once it's in the nucleus was kind of like a tumor promoter. Diabetes may be associated with disruption of GR-mediated glucose homeostasis. So like I said before, if it's playing a role in, if it's involved in glucose homeostasis and then you're inhibiting its function, then you might have diabetes. ED that acts by altering receptor function. So like I said before, once it gets to the nucleus, its function is changing, not its abundance. And then this is where I kind of started to get confused about. So the allosteric binding, so I was trying to like picture a receptor, and if DEX is still bound in the actual receptor, and so is like arsenite coming and binding like somewhere else on the receptor but not actually in its binding site. So it's like either, because an allosteric interaction, something like enhances or changes the way that it responds to something binding to it, right? Mm-hmm. Okay.
So it's binding at a site other than the hormone binding site.
Yeah.
I mean, they don't know that for sure. They're just hypothesizing this at this point. But whatever that allosteric, it's not affecting DEX binding, and it's not affecting the translocation thing, but it's affecting maybe its ability to bind to the GRE or something.
Or to like unmask the binding site for DNA or something like that.
Right. That's what I mean with GRE.
So glucose response